The main goal of this experiment is to produce ethanol from a glucose solution. This will be accomplished through the addition of yeast, which will consume the glucose and produce ethanol, water, and carbon dioxide. To simplify the process for this experiment, the yeast will be added to a new glucose solution, but it could also be done with the solution made during enzymatic hydrolysis if the antimicrobial agents tetracycline and cyclohexamide were not added to the enzymatic hydrolysis solution. During this experiment, observations of the fermentation process, including the frequency of bubbles over time, should be observed. After the experiment is concluded, the glucose concentration versus time data will need to be analyzed and interpreted. Both the change in glucose over time and the rate of carbon dioxide bubbles evolving from the solution should be slow at first. In the middle of the experiment, the yeast is multiplying quickly, so the glucose concentration should drop and carbon dioxide and ethanol production should be very fast. Towards the end of the experiment, the last of the glucose will be consumed and fermentation should slow down considerably. Rates of glucose consumption can be estimated from the slope of the tangent line at any point on the glucose concentration versus time graph.
For this experiment, you will need the following pieces of equipment: safety goggles, rubber gloves, heat-resistant gloves, a lab notebook, a pencil, a scale, a 100 milliliter graduated cylinder, two 250 milliliter Erlenmeyer flasks, a hot plate, a pressure cooker, an airlock, two rubber stoppers, the 10 to 100 microliter pipette, the 100 to 1000 microliter pipette, and a glucose meter with test strips. You will also need the following chemicals: yeast extract, glucose, distilled water, 70% ethanol, and yeast.
Make sure to keep an organized lab notebook. Record all dates, procedures, and results. Wear safety glasses, long pants, and closed-toed shoes. Also wear gloves to avoid contact with chemicals. Ethanol is highly flammable and must be kept away from ignition sources. Be cautious with the pressure cooker and the hot plate to avoid burns and electrical shocks. Handle glass beakers with care to prevent breakage. The instructions for this experiment are written with the assumption that you will be making a new glucose solution for fermentation. It is also possible, however, to use a solution made during enzymatic hydrolysis. As you may recall, during this reaction, enzymes are added to the pre-treated wood chips to convert cellulose into glucose. In a real biofuel production process, yeast would be added at the end of enzymatic hydrolysis, or even during enzymatic hydrolysis, and fermentation would begin. During fermentation, yeast will consume the glucose in the solution in the absence of air and produce ethanol, water, and carbon dioxide. The product we are interested in is the ethanol, since it is to be used as a renewable advanced biofuel.
Using the scale, measure out 1 gram of yeast extract and 5 grams of glucose. Make sure to zero the scale so as not to include the mass of the weighing dish in your measurement. Place these into an empty, dry, 250 milliliter Erlenmeyer flask, then use the graduated cylinder to measure out 100 milliliters of distilled water and pour it into the 250 milliliter flask.
A buffer solution, which will be used to dilute samples taken during fermentation so that they can be accurately measured by glucose meter, should be made. This solution will contain all chemicals in the fermentation solution, except for glucose, which is the independent variable. To begin making the buffer solution, first use the scale to measure out 1 gram of yeast extract and add to a 250 milliliter Erlenmeyer flask. Then measure out 100 milliliters of distilled water and pour it into 250 milliliter Erlenmeyer flask. Then use the scale to measure out 1 gram of yeast extract and add this to the flask.
Cover the tops of both flasks with tinfoil in preparation for the next step.
Both flasks need to be sterilized before the addition of yeast to ensure that nothing else is consuming the glucose during fermentation, like bacteria contamination from the lab. Sterilization should be accomplished using the pressure cooker and a hot plate. Conduct this step in a fume hood if available, otherwise on a lab bench. First, place the pressure cooker on the hot plate. Add 1 liter of water to the bottom of the pressure cooker. Then carefully place both flasks in the pressure cooker. Place the lid on the pressure cooker and turn to seal. Then wait for the water to come to a boil. The water has reached a boil once the safety valve, the knob on the top of the pressure cooker, begins to rock gently. At this time, the temperature should be reduced slightly, and then the flask should be left in the pressure cooker for 20 minutes. Watch the safety valve to ensure that it continues to rock gently, adjusting the temperature as necessary.
After 20 minutes have elapsed, shut off the hot plate and wait for the safety lock to release. This generally happens after about 5 minutes, and the pressure cooker cannot be opened before this time. Once the safety lock has released, remove the lid from the pressure cooker and remove both flasks. Wear heat-resistant gloves during this step, as the pressure cooker and flasks will be extremely hot.
Allow both flasks to cool to room temperature. This is extremely important as the yeast will be killed if the temperature is too high, and fermentation will be unsuccessful. Once both flasks are cooled, use the scale to measure out 20 milligrams of yeast and add it to both flasks. Swirl the flasks until a uniform solution is achieved.
Sterilize the airlock and rubber stopper with a squirt bottle of 70% ethanol. Spray all inside surfaces and allow to dry.
Fill the airlock to the max line with distilled water. Insert the assembly into the 250 milliliter flask containing glucose and yeast. Bubbles of CO2 should be produced throughout fermentation after startup period. The purpose of the airlock is to allow bubbles to escape and to keep other substances from entering, like air.
A bubbler could also be put on the buffer solution to show in the absence of glucose you won't observe bubbles because there is no fermentation taking place.
At the start of the experiment, take a sample from the fermentation flask to test for glucose using the glucose meter and test strips. The sample should be diluted using the following ratio: 960 microliters buffer solution per 40 microliters fermentation solution. Use a small plastic centrifuge tube for this dilution. The initial solution has such a high concentration of glucose, 50 grams per liter, that it must be diluted to achieve an accurate reading by the blood glucose meters. Your results from the glucose meter will need to be converted to grams per liter using the calibration curve. If you are using a new batch of test strips, remember to make a new calibration curve. If your results fall out of the range of the calibration curve, your solution may need to be diluted further. Remember, after your meter readings are converted to grams per liter, they will need to be multiplied by the dilution factor.
Allow the flask to ferment for 2 to 3 days at room temperature on the lab bench. Continue taking samples to test for glucose using the same dilution ratio as in step 9. Samples should be taken several times daily if possible. Record your glucose meter readings and dilution factor, 25:1, in your lab notebook throughout the experiment. You should see the concentration of glucose decrease over time as it is being consumed by the yeast. The process is complete when bubbles of CO2 are no longer being produced. Bubbles can most easily be seen by gently swirling the contents in the fermentation flask. Since glucose is being consumed at an increasingly fast rate, bubbles should also be produced more quickly as fermentation proceeds. Record observations in your lab book throughout the experiment.
After fermentation is done, glucose readings should be at 0. This is shown by a low reading on the meter. At this time, the contents of both flasks can be poured down the drain, if you choose to use the solutions for distillation. The airlock should be removed and a rubber stopper should be inserted in its place. Both flasks should be then placed in the refrigerator until you are ready to begin distillation.
Once the experiment is concluded, clean up the area and wash your hands before leaving the lab.
Convert your measured glucose readings by plugging them into the calibration curve equation made for this experiment, and then multiply by a factor of 25 to account for the dilution performed during sampling of the fermentation broth, in order to accurately measure the glucose concentration. Graph your data in Excel, with time elapsed on the x-axis and the concentration of glucose on the y-axis. The graph should have the dots connected by a curved line. Then calculate the slope of the line between the first 24-hour period, 24 through 36, 36 through 48, and 48 through 72 hour period to show the differences in the rate of consumption of glucose at different points in the fermentation. At time 0 and time 72 hours, or your ending time, consumption rate will be at 0. The rate will slowly increase until 24 hours, then explode and increase at a high rate until 36 hours, where it will reach its max. Then the rate will still be very high, but slowly decrease until 48 hours. At 48 hours, the rate will decrease substantially and then decrease at a slower rate until it reaches 0.